So the folates are a group of activated vitamins. Again, folic acid is an inert substance, but when it goes on to become methylene or methyl or 5-tetrahydrofolate, methyl tetrahydrofolate, these are the active forms of coenzymes on them. There's a lot of talk now, particularly in America, it's known as B9. In some textbooks in Europe, it's actually called B11, which makes it more confusing. We prefer to call it folates because then it's not misconstrued, and it's possible from some probiotic manufacturers that you can get that it may be that folate is actually being able to made by certain bacteria in the gut. So in a sense, this is why folate was never given a number, because it was actually managed to be synthesized in the body. So if we just call it folates, that covers the whole family there. So we get it from folic acid, which then goes through this reduction process and always has hydrogens added. First of all, to make dihydrofolate, and then tetrahydrofolate. So the H4 on here is tetrahydrofolate. So this has 4 hydrogens added to it, which uses NADPH here, the active form of vitamin B3. So what we've got here is tetrahydrofolate, and this itself is actually an active coenzyme. Now, what we do is we can add then a CH group to it, a CH2 and then a CH3, the methyl form. So the first form in this pathway that we add here is to methylene tetrahydrofolate, which is CH2. So CH2 is methylene tetrahydrofolate, and the methyl donor here is given by serine, which converts round to the amino acid glycine. So in this pathway called a methyltransferase, it donates 2 methyl groups. So you'll see that that's pyridoxal 5-phosphate and zinc dependent. So here we see that link that I talked about before, that B6 is influencing folate and B2 influences B6 and so on, back in the chain here. Now that conversion, that production of the methyl group to methylene is able to actually act the methyl group to uracil and to convert it into thymine. And this was the first polymorphism that was discovered about something like 20 years ago, was when you got a U or a uracil into the thymine position. So it's like having an RNA component in the DNA, and this became known as a single nucleotide polymorphism. In other words, a U into a T polymorphism. And this encoded, they found that led to a deficiency of methylene tetrahydrofolate, and by supplementing methylene tetrahydrofolate, it resolved the polymorphism. And this was the first polymorphism that was discovered to be linked to breast cancer. So hence the link with folic acid, or folates more specifically, in helping to prevent the incidence of breast cancer. Methylene, or CH2 tetrahydrofolate, they can have another methyl group added to it to make the methyl tetrahydrofolate here, which is CH3. So technically this is called 5-methyl tetrahydrofolate. And this enzyme here, this methylene tetrahydrofolate reductase enzyme, again is one of the more common polymorphisms that we come across. And these are people who require methyl tetrahydrofolate as a supplement because they can't activate this enzyme sufficiently. So these people would weaken to methylene tetrahydrofolate and strengthen to 5-methyl tetrahydrofolate. So it's the same basic functional biochemistry that we talked about before. On this side, we can have 4-methyl tetrahydrofolate, which is otherwise known commercially as folinic acid. And this converts with the addition of water to methanol, or CH. So methanol is CH, methylene is CH2, and methyl is CH3. So this can go on to make methylene tetrahydrofolate also. So some people will respond to folinic acid, or CH, some to methylene, and some to 5-methyl tetrahydrofolate, just depending upon which of these enzymes doesn't function correctly. So we see that the methylene, the methyl tetrahydrofolate, basically its function is to donate a methyl group to cobalamin, or B12, to convert it into methyl cobalamin, which then converts on to recycled homocysteine that we'll see in a minute. So it sounds complicated, but the ones to remember, the biologically active forms of folate is tetrahydrofolate, methylene or methanol, methylene is CH2, and methyl is CH3. So these are basically the functions here. The methylene function is necessary for the formation of cytosine, adenine, and methylation of uracil, which I mentioned there. It's necessary to methylate uracil into thymidine. The 5-methyl is necessary to recycle homocysteine, and the cobalamin, methylcobalamin, is necessary also in the synthesis of adenine and methanol necessary in the synthesis of adenine and guanine for the DNA. So folic acid is basically a synthetic, not activated form of folate. Richer sources are liver, and the key here is dark green leafy vegetables. The word folate means foliage, comes from the word foliage, so anything with a dark green is a good source of folate. Beans, wheat, germ, yeast. Muscle testing tends to indicate that cabbage is the best. Cabbage seems to be one of the best sources of folate. Other sources include egg yolk, dairy, beetroots, etc. So it's important that we have sufficient intakes of it to build our proteins and our DNA for the next generation of cells. So it's involved with the formation of blood cells, ensures rapid cell growth. So every time a cell divides during mitosis, every time a cell divides into two cells, it needs 2 molecules of folic acid, or folates more specifically. So a deficiency of folate will lead to an inability to divide the cells or to a mutation in that cell because it won't develop properly. Okay? So if it's a blood cell, then you end up with an anemic type of condition. If it's a malignant cell, or it could change malignant because it doesn't divide properly. So European Food Standard Safety Authority allows you to say that it's normal blood formation, normal homocysteine, normal metabolism of the immune system, normal cell division, normal maternal tissue growth, normal amino acid synthesis, and psychological functions. So they're all normal, if you like. So they know that low levels can create abnormal, but they can't say, or you're not allowed to say, that folate will actually improve if you are in those states, in any of those, but we know that it is. So folate deficiency can result in inadequate intake, defective absorption, or increased requirements. So the early symptoms would be tiredness, irritability, and loss of appetite. Severe deficiency will lead to megablastic, which means a bigger than normal red blood cell, which is immature and oversized. Pregnant and breastfeeding women are at higher risk. So again, very, very important during pregnancy to take extra, but it has been shown that it's better to take more of the 5-methyl tetrahydrofolate in case the person can't convert folic acid. So it's not a good idea to increase folic acid because that may actually limit the amount that they can convert to the 5-methyl. So it's better to give the 5-methyl, which can then go either way and increase the methylene, etc. So the intestinal cell has on the surface, a brush border, a specific folic acid binding protein. It transfers folic acid to the inside of the cell. Here the dihydrofolate reductase reduces the folic acid to tetrahydrofolate. So most of the activation actually occurs in the intestine in this particular time. Methylation then occurs either directly or indirectly to make the methyl tetrahydrofolate. It's used, as I said, in the production of the different purines and pyrimidines. Most folate in the serum is in the form of 5-methyl tetrahydrofolate. Serum folic acid levels decrease in pregnancy, reaching their lowest at full term. So because, of course, the baby's growing fastest at that time, more cell division, and so you need a greater amount of folic in the last trimester. Look out for post-pregnant cervical dysplasia. So cervical dysplasia means the beginning of erosions of the cervix, which is quite common after pregnancy because the level of folate is down. And this goes usually with dysplasia in the gums. So here we see a brighter reddish gums and usually receding teeth. receding gums, bigger teeth. So when you see this, think folate. Think usually 5-methyl tetrahydrofolate as opposed to folic acid. But you must act on that quite quickly, because more people lose their teeth from periodontal wasting gums than from dental caries these days. So you just open the mouth of the person and you look for receding gums. Sometimes they're red, sometimes they're not, and just therapy localize it. Strong muscle goes weak, and then you find that it responds to one of the folates. All right, so that's the most common symptom that you see is receding gums of folate deficiency. It's the methylene form, the CH2, which is the active form of folate, which helps prevent neural tube defects. Again, every time a cell divides, it needs more folate, and of course, if it divides in that early embryologic stage, it could lead to a spina bifida and possibly other compounds if the mother doesn't have enough methylene tetrahydrofolate. So we saw it functions in nucleotide, homocysteine and glycine synthesis. We mentioned about the anemia, normal DNA replication. We've talked about the 4 biologically active coenzymes there. Deficiency symptoms could lead to birth defects in pregnancy. So very, very important, of course, to take the folate before you're pregnant. Even the whiff of thinking about being pregnant is very important to take extra folate, okay? Because once you are pregnant, you don't know you're pregnant for at least probably 2 weeks. Most of the work has already been done. You know, the baby's then forming the placenta after 2 weeks. You're forming the placenta and the baby's cells are coming out of that zona pellucida, and then it's too late almost, because you've already done the mismatch of any genetic makeup. So it's very, very important prenatally to take 5-methyl tetrahydrofolate rather than just folic acid, in case you're one of those who can't activate it enough. But the easiest way is to inspect the gums. It's what they do with horses. So when you have a look at a horse, you know, the vet always looks at the gums and sees the state of the health of the horse, if they're suitable for breeding. And it's the same with humans. If you've got a potential daughter-in-law, you need to go along and check the gums for a good stock. But retarded growth, classic of children who don't grow, okay? Children who don't grow, like our parents said to us and their parents said, if you don't eat your greens, you won't grow to be a big boy or girl. And that was very true. Yeah. So where do you test this? You test it on the epiphyseal growing plate of the long bones. So you therapy localize around the epiphyseal plate of the tibia or the inferior part, the distal part of the femur. Therapy localize down. You get a strong muscle goes weak into a small boy or small girl, and then you test them with the folate, and if they go strong, you know they need more folic acid or folates. And the mother or father will always tell you they don't eat green vegetables. You know, this is the problem. They just won't eat them, and so you've got to use it as a supplement. Scaling dermatitis, alopecia, loss of hair. Of course, this is a game where you see it when you block folate activation with methotrexate and other drugs and chemotherapy as a result. The first place you start to see side effects is when the hair drops out, because this is where we grow and we need more cells. So the first place you see that inability to produce new cells is in the hair, and so unfortunately, you know, with most of these folate blocking agents. The trouble is it's a dilemma. Does the person with the cancer, who's had the cancer treatment, need extra folate to stop the mutation from being formed? Yes. If, on the other hand, you feed them the folate, do you add to the multiplication of that? The jury's out, okay? So some people will say, well, the person needs more folate to stop the mutation, and other people will say, well, we're trying to block the folate and the ability to do it, so we can starve the cancer from reproducing. It's difficult, isn't it? If we knew the answer was to prevent it in the first place, is to make sure you get enough folate. Because certainly 5-methyl tetrahydrofolate shows up with most cancer cases. Not all, but with most cancer cases. Does not mean that everybody who has strengthens to 5-methyl tetrahydrofolate has cancer, okay? Because there's plenty of other symptoms. But we do find, of course, cancer is involved with abnormal mutations in the gene, and usually requires more folate to correct it. So megaloblastic anemia, these means bigger cells than cells which carry less oxygen around. Cervical dysplasia, we talked about, particularly post-pregnancy, mouth ulcers, in maybe 5%. And depression, of course, because it's involved with a lot of neurotransmitters. Dr. Goodhart says in cases of bad hematoma, that's bruising, use pancreatic enzymes and folic acid. 90% of folates are lost with steaming vegetables, hence the importance of drinking the vegetable water. So when you cook vegetables, you should always drink the water, make it into a nice gravy, etc. 70 to 90% are lost with frying in an open pan. Wally Smith says yellow tongue is the need for folate. Rather than saying folic, we'll say folate now, I think, and certainly Wally, who's a great advocate of 5-methyltetrahydrofolate, I'm sure would agree there. He finds in his practice that's the most common polymorphism where he is. In pernicious anemia, there's a rise in serum levels of methyl tetrahydrofolate and an increased excretion of forminoglutaric acid or FIGLU. Check this especially in MS cases. That's our picture of the recycling of homocysteine. Dr. Goodhart says take up to 10 to 20 milligrams, that's quite a hefty dose of folate, during attacks of gouts. Might be worth remembering that. Personally, I always find zinc also very, very important. Plenty of water and sodium bicarb to alkalize the system. That's what we find in here. But George always put down high doses of folate. So folate deficiency, clinical expression times of homocysteine. Homocysteine is one of the first blood tests which shows up folate deficiency. So homocysteine will rise within 10 days of having lower than optimal levels of folate, whereas serum folates, urine excretion of FIGLU, megaloblastic marrow anemia take a long time before they actually show. But high levels of homocysteine will occur within a couple of weeks. So homocystinemia is the best functional marker, in fact, for folate. So if a person weakens to homocysteine, first thing to check is the folate. Sources we've talked about already, with cabbage being the number one there. Daily recommended allowances vary tremendously. Obviously, pregnancy, we need substantially more. And interestingly, in David Walsh's book, there's no specific muscles for folates, although I'm sure we can develop plenty of biochemical pathways to indicate this, starting, as I say, with homocysteine. Ready, Gary?
Ready.
So this slide shows the different enzymes. It's a little bit complicated, of course, but if you actually look at it, you can see how the different folates are converted. We've talked about tetrahydrofolate. You probably need to blow this one up to be able to see it. And we talked about methylene, the CH2, and the methanol, the CH tetrahydrofolate, and how they all interact. So basically, where we've got arrows going both ways, one can convert into the other one. And when you've got an arrow only going one way, it means it's a one-way traffic only to that. So the one where the one way, you've actually got to take the preformed amount, preformed substrate, because it can't be made from other forms of folate. So that one's worthwhile looking at in a bigger way. You don't have to test for each of these, but you can soon see that if a person shows to 5-methyl tetrahydrofolate, then basically you can see that it's made from methylene tetrahydrofolate here, and will go on to recycle homocysteine. So it's a nice picture, that one. I might blow that one up and give it to you to put into your folders there. This is how we break down basically alcohol, which is ethanol. Ethanol goes on via alcohol dehydrogenase to make aldehyde, or specifically in this case acetaldehyde, which then eventually will break down to acetic acid and carbon dioxide and water. Whereas methanol, which is actually produced from abnormal bacteria, bad bacteria in our gut, instead of fermenting good ethanol, will present and ferment into formaldehyde, which is much more toxic substance, which then goes through the same enzyme, the aldehyde dehydrogenase, to form formic acid, which is what we talked about, red ants kindly give you and makes you itch. And then that formic part goes round with folic acid to make the formyl tetrahydrofolate, or folinic acid as it's called. Okay, so we actually produce folinic in this pathway. Formic will then again degrade eventually to carbon dioxide and water. So here was the example of ethylene glycol, which is antifreeze, isn't it? the breakdown of that, if that got into your body, goes through the same enzymes, aldehyde, dehydrogenase, and then blocked down to oxalic acid and oxalates in the renal tubes. This is just our press research that too much synthetic folic acid may interfere with the absorption and utilization of bioactive folate. So this is why they suggest that you don't take high amounts of folic acid, because it actually inhibits, if you take a lot of the substrate and you've got problems synthesizing that into the active forms, you may actually slow that pathway down. So I think that's something that we need to consider with all these vitamins, the B vitamins, is you need to test with the biologically active form. So in this case you need to test with the folates, and then if they strengthen that, you can go back and test them for folic acid. So if a person does strengthen the folic acid, then they are deficient in folates, so they can be deficient in them all. If you give high amounts of folate and they don't need it, they may not be able to make it. And if you give equally high amounts of the made stuff, the activated form, and they're actually deficient in folic acid, you'll inhibit that conversion. So this is why, but you do find that, in my experience through the years, very few people are deficient in folic acid. Very few people are deficient in B6, surprisingly, but heaps of people are deficient in pyridoxal 5-phosphate and 5-methyl tetrahydrofolate. So it's that enzyme that converts the two. Okay, so we're going to move on now to vitamin B12, otherwise known as cobalamin, and the cobal stands for cobalt, because right in the center of the molecule, which is very similar to the molecule of heme, instead of having an iron in there, we've got cobalt in there. So the main function of B12 is to be converted into two biologically active coenzymes. One's called methylcobalamin, which obviously has a methyl donated to it, and the other is deoxyadenosylcobalamin, which is generally shortened to adenosylcobalamin, which has the adenosyl group donated usually by ATP. So here we see the conversion of hydroxycobalamin, which is the raw vitamin B12, if you like. Hydroxycobalamin is straight for B12, which is what you'll get from your diet, from animal products. And then in the body, that'll convert to methyl or to adenosyl. The prime function of the methyl is to convert homocysteine back into methionine, so it recycles homocysteine in that pathway. And the adenosyl, as we see, is more to do with maturing red blood cells and burning odd-numbered fatty acids. So it's the largest and most complex of all vitamins. Doesn't mean largest, it means largest in size, the molecular shape, not the amount. Comprises the only cobalt-containing molecule in the human body. It's produced exclusively by microbial synthesis in the guts. Now there is some produced in the human gut, but it's a little doubtful how much we can absorb and whether we have that particular type of bacteria. But ruminants are generally the animals which do this. So they have particular bacteria in their gut. In other words, they recycle grass and things like that and redigest it in ruminant cases. And they have specific bacteria that do that, that actually make the B12. So it's the bacteria that make B12, not the animal. The animal doesn't make it, it's the microbes that live in their gut. So animal protein products, particularly organ meats, like liver and kidney, lamb, etc., are the good sources of B12. In fact, we find that lamb, again, is one of the richest sources of so many of these, and because lamb is generally not put onto grassy fields, which have herbicides because they live on hillsides, and they will eat most things except thistles and stinging nettles. Here it says other good sources according to the books of fish, eggs, and dairy products. Kinesiologically, this is not true. I haven't yet found a person who's really strengthened to fish, eggs, or dairy products who's B12 deficient, because it's not red. It has to become from basically red. That's the cobalamin part, is the red meats. So red meats are the major source. You may get small amounts from the other ones, but it is small and maybe insignificant amounts for people who don't eat red meat. So sufficient intake of B12 is important to help the body convert food into glucose, to maintain healthy nerves, and produce the different nucleic acid DNA building blocks. It's regulated together with folates to form red blood cells, And sometimes you need to look at both because they have a certain overlap of one can achieve the same as the other in treating anemia. So megaloblastic anemia can be from low folate or low B12, but the neurological symptoms are B12. So there is that risk that if a person has a megaloblastic anemia, that they're given folic acid, that they may actually end up with neurological problems because it's only the B12 which solves the neurological part. So you have to be careful with that. European food safety standards say that it's involved with red blood cell formation, very true, normal cell division, energy metabolism, immune system, homocysteine, neurological, and reduction of tiredness. Certainly the most important neurological one there is the myopathies, where you get loss of sensation and sometimes initially pain, pins and needles or paresthesia in the legs, but particularly the feet, and then eventually loss of sensation in the feet. So people who lose that, it's usually B12 deficiencies. Mild deficiency is not uncommon in elderly people. 10 to 15% of injured was over the age of 60. Other research that I've studied is in 80-year-olds. It's something like 90 to 95% of 80-year-olds are deficient. So this is really the thing that is involved in maturing the red blood cells, and therefore the red blood cells go on to carry the oxygen. So a lot of elderly people are basically deficient in oxygen, which leads ultimately to dementia. So this is really dementia rather than Alzheimer's dementia, just due to hypoxia or lack of oxygen in there. Low levels can cause a group of symptoms from shortness of breath, all the classic symptoms of anemia, of course, nervousness, numbness, we said, neurological damage. We said it's produced in the ruminants of animals by bacteria. It needs intrinsic factor, which is a protein in the stomach, which is secreted when we secrete hydrochloric acid. So in the oxyntic or parietal cells, when we secrete hydrochloric acid to start to activate pepsin to digest proteins, we also secrete intrinsic factor, which is a peptide which locks onto the B12 in the stomach and carries it through the intestine to the terminal end of the small intestine, where it's absorbed, intrinsic and the cobalamin, at the end of the small intestine. So it has to have the intrinsic factor to get absorbed. So therefore, if you have low hydrochloric acid, if a person suffers from hypochlorhydria, they're not going to produce intrinsic factor in sufficient amounts, and therefore they get B12 deficiency. So a person can get B12 deficiency, not because they're not eating red meat or the sources of it, but because they're not producing the intrinsic factor. So the critical thing here is that people should take B12 if they need it as a supplement when you produce acid. And the time when you do that is obviously at stomach time, which in standard time zones is 7 till 9 in the morning, so it's breakfast time. So B12 should always be given with food because it then stimulates the hydrochloric acid, which in turn will then stimulate the production of intrinsic factor, which will help you absorb it. So very, very important, the timing there. Conversely, folates actually don't like acid. You know, when you try and dissolve folate, it doesn't dissolve in a normal pH. You have to actually alkalize the water a little bit in order for folate to dissolve. So the optimal time to take all forms of folate really is last thing at night when the level of the stomach acid is lowest. So just before going to bed, if you prescribe folate, 5-methyl-tetrahydrofolate, folinic, etc., it's better to then give that last thing at night unless the capsule is in a delayed release form, so it bypasses the stomach for the acid there. For a long time, they thought that the intrinsic factor was actually pyridoxal 5-phosphate, but it appears to be a peptide which requires pyridoxal 5-phosphate for its activation. So there's this link again back with pyridoxal 5-phosphate and stomach acid, which we talked about before. So hypochlorhydria is associated or can lead on to B12 deficiency. People with intestinal parasites, especially cestodes that come up the small intestine, like Diphyllobrothium latum, may develop B12 deficiency as the worms live in the lower part of the ileum and compete for the B12. So these are cestodes, or tapeworms as they're colloquially called, actually come up from the small intestine, the large intestine through the ICV into the small intestine, and they'll be there ready for the B12 because it's being nicely carried down by the intrinsic factor and only absorbed in the terminal end of the small intestine or the ileum there. So here we see cobalt in the middle there. So cobalt is a blue colour here, as you can see, cobalt blue, like when fireworks go up there. And it has a strong affinity, cobalamin, to cyanide. It binds onto cyanide, so it's often used as a detoxifier of people with cyanide toxicity. So often, when it's bound on there, it's called cyanocobalamin. So as a supplement, cyanocobalamin can give you the cobalamin, but it does give you a dose of cyanide. And if you're a person who's sensitive to cyanide, particularly like people with isothiocyanates, like sensitivity to broccoli and other green vegetables, certainly you don't want to administer cyanocobalamin. Hydroxycobalamin is the better source because that's neutral, and that can take cyanide if we've got an excess cyanide, which is common, in our mitochondria. When we methylate it, it's called methylcobalamin, and when we add the adenosine, it's called adenosylcobalamin. So those really are the different forms of B12. So functional test really, as we'll see in a minute, is the deoxyadenosine. Adenosine, basically one of the important functions is to convert and help burn odd-numbered fatty acids. So the body doesn't normally use odd-numbered fatty acids very well. So we use even-numbered, and these are saturated fatty acids. So we start with carbon 2, 4, 6, 8, and then we get up to carbon 16, which is palmitic, and then carbon 18 is stearic, and so on. These give the strength to our cell membranes and to our myelin and things. Odd-numbered carbon fatty acids don't give the strength, they don't integrate in the way. And these are propionic, valeric, etc. types of fatty acids. And when we build these up, these can be actually quite toxic, particularly to the myelin sheaths. So this is important in people with demyelinating diseases or neurological problems. So B12 helps us to burn the propionic, which is a 3-carbon, into the Krebs cycle. So a strong muscle here would weaken to one of the intermediates in this called methylmalonic acid. And the methylmalonic acid test is a standard lab test for people with B12 deficiency, much more than measuring cobalamin, because cobalamin itself is linked to a protein and carried around called transcobalamin. So it actually doesn't tell you what's happening inside the tissue. Whereas if a person has a high level of methylmalonic acid, this is indicative of adenosylcobalamin deficiency. So this is the actual diagram taken from Harper's Biochemistry. So here we've got carbon 3. See, we've got 3 carbons here, fatty acid, propionic. Okay? Now this is made in the gut with a lot of bad microbes. So when people have a bad, dysbiosis, they tend to make these short-chain fatty acids, which should normally be carbon 2 acetic or butyric carbon 4. These are good for the colonocytes to feed them, but carbon 3, 5, 7 are not good. And these are produced mostly by bad bacteria in the gut and then get absorbed. But this pathway also burns certain amino acids and is able to, like these branch chain ones like leucine and valine, also go into this pathway. So this pathway, propionic, goes along around here to a substance called methyl malonyl-CoA, and then is shunted here in the presence of B12 adenosylcobalamin to making succinyl-CoA, which is part of the Krebs cycle, as we'll learn in later modules. So this is how we burn these odd-numbered fatty acids and certain amino acids. So if we're deficient in adenosyl, which is usually because we don't activate it, genetically it's a polymorphism here, what happens is we get a build-up of methyl malonyl-CoA, which goes on to make methylmalonic acid, which I said is the standard lab test for B12 deficiency. But in blood kinesiology, of course, we can use methylmalonic acid as a marker. The person would weaken to that, and what we find is they would strengthen to succinyl-CoA, because that's what they need. That would be the end product. And this enzyme, methylmalonyl-CoA isomerase, or mutase as it's called, may have a genetic polymorphism in it, which is what we find a lot. That maybe people only express 50-60% of there, and so they get this backlog or build-up of propionic acid, which leads to a lot of neurological problems. So we'll discuss about all those later on, and particularly in relationship to neurological problems. So I like this picture here, this process, because a lot of people do show with neurological problems to carbon-3 propionic excess. So deficiency leads to accumulation of propionic and other odd-numbered fatty acids. Bacteria in the gut of ruminants produces large amounts of propionics, which are then absorbed and get into the intestines. That's bad bacteria, mainly the gram-negatives. Consequently, intake of animal and dairy products results in higher levels of odd-numbered fatty acids. A similar process occurs in humans who suffer from dysbiosis, which is gut imbalance there, get a lot of wind. The association between B12 and abnormal fatty acid synthesis, those are odd ones, provides a rationale for the neuropathy of B12 deficiency. So this is why so many people do respond who've got neurological problems to vitamin B12, and sometimes they only by injection does it work, because to bypass the stomach if they've got intrinsic deficiency, which would lead on to then pernicious anemia because they can't absorb B12 from the diet. So these people have to have B12 by injection. But most of our side clinically are people who are unable to, they get enough B12 but they can't convert it into the adenosine or the methyl. Odd-numbered fatty acid chain fatty acid build up in the central nervous system lipid membranes with B12 deficiency, resulting in altered myelin integrity and demyelination. That's why one of the first things with multiple sclerosis and demyelinating disease is to check the B12. And many respond, quite well, and they feel much better when they take B12, have B12 injections, or better still, when they take adenosine cobalamin. So a functional test for methylcobalamin would be homocysteine, because again, methyl gives its methyl group to homocysteine to recycle it. So in this case, a strong muscle weakens to homocysteine. And as we've seen, the B12 gets its methyl group from methyl tetrahydrofolate in turn. So here we've got cobalamin going on to make methylcobalamin, and it's donated by methyl tetrahydrofolate. So that's back to our one carbon cycle again there, which we've seen quite a lot of, and we'll see it again with S-adenosylmethionine a bit later on. So adenylcobalamin deficiency, hypoxia, fatigue, and ultimately dementia because of lack of oxygen in the brain. So this is when people behave quite peculiarly, but actually haven't got the amyloid protein plaques and things, other indications of Alzheimer's disease. Methylcobalamin, as I say, is more involved with central nervous cardiovascular disease, pernicious anemia, and homocysteine recycling. So that's really all you've got to remember. The two types work differently. Adenosine helps us to burn these odd-numbered fatty acids, and the methyl recycles homocysteine. We talked about bacterial synthesis in ruminants, but it does occur in humans, but as far as we know, very little is absorbed. It would be good if we had those bacteria, because then we wouldn't have to eat red meat if we didn't want to eat red meat. But they haven't mastered exactly which bacteria it is. I've seen articles about different types of bacteria in animals that can do this, but they're not natural bacteria to humans. And so if you take these as probiotics, they don't stay. They come on through. So there's a lot of interest in probiotics of animals, especially cow dung ones, especially camel dung ones, but they're not natural bacteria to humans. So they do wonders when they're going through in transit, but they don't have a lasting benefit. So really the best probiotics are the ones that normally do reside in the human gut. Dr. Godard says, don't forget to give the patient B12 with fascial agglutination problems. As you know, if you get the fascia bound down or glued down to the surface of the muscle, when you stretch the muscle, it doesn't want to stretch. So the fascia and the muscle get bound down, so you don't get that nice gliding sort of motion. So the test for a fascial problem is you test the muscle, and then you stretch the muscle, come back and you retest it, and it goes weak, which means that the fascia and the surface of the muscle are stuck down or occluded. And he now, in his latter time, used to use the percussor a lot to do this, to break the bonding down, the fascial agglutination. But B12 is very important for these sort of problems, people with recurrent fascial. This is really the start of things like rolfing, you know, these techniques where they really massage the fascia off, myofascial techniques of massaging the fascia off. It can be very painful, especially with brain-damaged children. Dr. Goodhart said use B12 and tryptophan for depression. So we would look a little bit more probably into that tryptophan serotonin pathway now, and look perhaps for supplementing maybe 5-hydroxytryptophan or B6, B12, etc., to activate the conversion of tryptophan into serotonin. Dr. Goodhart says use B12 for painful and recurrent trigger points. Again, very similar like fascial problems. Trigger points, when people develop a lot of trigger points in their muscles, think B12. Sources, anywhere where we store, we store up to 3 months or more of B12 in the liver, and therefore liver is a very good source of B12. Meats, especially lamb and other red meats. And then gradually, as you go through, as we said, poultry, fish, eggs, dairy, and really we're getting down there to such minute amounts. Recommended daily allowance 6 micrograms. This varies. Some books, European ones, say 1 or 2 micrograms. 6 by the FDA is quite a high amount, but really, in people who are deficient, they need a full milligram or 1,000 micrograms. So they need a lot to get them going. Sometimes people find they have a hefty dose, and then they only need that once every month, to take a hefty dose, because the liver stores it and then uses it out. Other people find, they need their, let's say 100 micrograms, 1,000 micrograms on a regular basis. We're all very individual, and it depends on the conversion there. I've seen a lot of amazing effects with people who take adenosylcobalamin, which they don't get with just ordinary cobalamin. Psychiatric problems, neurological problems, incredible differences sometimes. Muscles associated with B12, David Walther doesn't come up with anything specific. I've tried here thinking, well, maybe dementia's involved here, hypoxia, could be the lung meridian, could be the conception vessel, because that's related to supraspinatus, which could be related to the brain function. Okay, so there's 2 muscles associated with the conception vessel. There's the supraspinatus, which does the first 30 degrees of abduction of the shoulder joints, and the one that most people forget is the diaphragm, which is in the midline here. So the diaphragm itself is operated by the phrenic nerve, but meridian-wise is the conception vessel. So let's do the supraspinatus. So elbow straight, slight abduction there, and what you're going to do is you're going to push out and slightly up, all right? In other words, not straight out, because the scapula comes round, and therefore, if you push, keep the elbow straight, that's it, and push out and up, you see we've got a nice weakness there. And again, 10-20 degrees of abduction, push out and slightly up. So we've got the weakness of the supraspinatus muscle there. So what we're going to look for now is a little bit of see whether B12 will help you. So what we do is, first of all, we'll do the adenosylcobalamin. So we can use the muscle as our indicator. Does she strengthen to the muscle? The diaphragm, by the way, is a muscle very difficult to test. We did it with B1, if you remember, by the breath holding. But the easiest way is to actually therapy localize the insertion of the diaphragm around the lower costal margins, about 7 and 8 there, just on the one side. And then to test here. And then you can do the other sides, because we basically have 2 diaphragms. We have 1 on each side. And we can see that we're getting a positive on that as well. So let's pop a few granules of B12 on. Okay, don't swallow. That's the adenocyl. So let's get in position and push out. Beautiful. And elbow straight and push out and up. Good. And let's do the diaphragm as well there. So we can see it strengthens the diaphragm as well. Pull, pull, pull. Good. So, do you get breathless? If you exercise, yes. So possibly you get breathless a little bit more than what you should do, because the more you can improve your oxygenation, the maturation of the red blood cells, the better oxygen you start to carry around. Okay, thank you.